This recorded lecture will take the place of the lecture that we normally have on Fridays. Today is Friday, January 31st, and class is canceled, but I wanted to make sure that you still had the benefit of some explanation of the material that you're responsible for. We'll first go through the schedule. We are in the electrical signaling in neurons week, and we're just finishing that up. So on Monday at noon, you're going to have your ion channel quiz due. That one was put up yesterday morning, so you should get started on that. You'll have one more quiz on synaptic transmission, which we'll cover next week before your exam, which will be on Friday, February 14th. There are lots of study resources being uploaded, so you should take a look at those if you're worried about the exam. There's also a possibility of us doing, say, a tutorial session or a review session the week of February 10th, but we can talk about that in lecture next week. So today we're going to cover membrane potential in a little bit more detail, and we're also going to talk about the action potential and how axons propagate signals down the length of the axon and how those signals are insulated using myelin. First, I wanted to show you the giant squid. So this is an animal that has been exploited in neuroscience because of the size of its axons. So if you're a neuroscience person, you've seen giant squid axon referred to in the Hodgkin and Huxley experiments, but I wanted you to see what this thing looked like. So the giant squid exists mostly in warmer waters, but they are very, very deep in the ocean. This is one on the right that washed up in New Zealand, and you can see the size of this thing. So let's just take a look at the difference between an axon and a nerve. Now, a nerve is a bundle of axons and dendrites in some cases, but what they do when they do these experiments in the giant squid is they take out this stellate ganglion here, and a ganglion is a bunch of cell bodies for neurons, and these cell bodies in the giant squid have axons that extend all the way down to the end of the animal, and these nerves contain the axons. So just to put it in perspective, this picture here, this is a surgery that was done on somebody who had sciatic nerve impingement. So this person's sciatic nerve is right here, and you can see that this is enormous. This sort of comes out and runs down the back of your butt. Within your sciatic nerve are all of the axons that are sending information to your leg. And we'll look at the anatomy of that when we do spinal cord anatomy. But for now, just know that when you look at a nerve, this is many, many thousands and thousands of axons bundled to make this nerve. So I found a video which I thought was interesting. Why don't we take a look at it? This is a video that has Alan Hodgkins in it, and he's measuring membrane potential from the axon of a giant squid. And you can see that it's a little bit of a misnomer. The giant squid is giant. The axon is giant relative to some axons, but it's not very big. So let's take a look at that now.
Placed inside the axon by Hodgkin and Huxley in 1939 at Plymouth, and by Curtis and Cole in 1940 at Woods Hole. Hodgkin and Huxley first made a plastic cell and mounted it on a platform which could be raised and lowered like a lift. As Professor Baker shows, the axon held by the cannula was then hung in the cell, which was filled with seawater and connected to an external electrode. The internal electrode, which was not attached to the lift, was then placed vertically above the axon, with its tip in the cannula.
So just to be clear, if you haven't done electrophysiology before, and I would imagine that most of you haven't, what you're looking at here is called the cannula. The cannula is glass, and what you do to be able to get it into an axon, which is really, really tiny, you put the cannula into a machine called a puller, and it creates a current that heats up the glass in the middle of its length, and the puller pulls both ends and creates basically like a super, super fine needle out of this cannula. So you use the glass cannula. It's not an electrode per se, but you use the glass cannula to stick it into the membrane of the axon.
It was centered with respect to the cannula and axon by placing a small mirror beside the axon, arranged so that a second image at right angles to the first was seen through the microscope. By adjusting the position of the cell, both horizontally and vertically, the cannula and axon were now raised up over the tip of the electrode, which always remained in the same line of sight. Hodgkin and Huxley found that as the electrode entered the axon, a negative potential with respect to the external seawater of about 65 millivolts was obtained. This was the resting potential of the axon, and although its existence had long been suspected, this was the first time it had been directly measured.
So here you can see their measurement of minus 65 millivolts. And at this point, he was putting the cannula into the axon, and you can see the voltage inside the cannula decreasing because there's an electrode inside the cannula. And then it holds because he's just measuring the voltage between the outside of the axon and inside the membrane of the axon. So it's just going to hang out here over time because he's not doing anything to that axon.
Moreover, when the axon was stimulated, the action potential did not simply fall to zero during the impulse, but became positive with respect to the outside, shown by the overshoot of the action potential. This important discovery suggested that the nerve membrane, which at rest is mainly permeable to potassium, becomes primarily permeable to another ion during excitation.
Now we know, because we've already done this, that this depolarization away from negative values inside of the cell is due to the opening of sodium channels. And then the repolarization and subsequent hyperpolarization of the cell is because of potassium channels. This here is the voltage that they're injecting to stimulate the neuron, and it's sort of opposite to what we would think. But the flashing on the screen is important, because what he's doing is he's measuring only in one single teeny tiny space on that axon's length. So what he's seeing is stimulation of the channels and a change in membrane potential only in that region. I think that because these things are drawn, and sometimes drawn really slowly, it implies that this line is the current traveling down the axon. But in fact, this is one place in the axon, and in that one place, the membrane becomes depolarized, hyperpolarized, and then slowly goes back to its normal resting membrane potential. So we've looked at this already, but I want to remind you that that resting membrane potential is maintained by many different types of channels, specifically by a sodium channel. This is a leak channel that's always open and allowing some of the sodium to pass into the cell. There are potassium channels that are also leak channels that are opened all of the time. And there's the sodium-potassium pump that requires the ATPase to move 3 sodiums out of the cell in exchange for 2 potassiums moving inside the cell. And this is what maintains the difference in charge so that the outside of the cell is more positive than the inside, thereby giving it the resting membrane potential of about minus 70 millivolts. Remember, though, that there are also other types of potassium and sodium channels that you read about in the reading that are voltage gated, that inactivate and activate at different membrane potentials. So this is an experiment that you also would have read about, which is called a voltage clamp. And when depicting a voltage clamp experiment, what we typically do is we show the voltage that you're forcing the membrane to be, and then you're showing the current underneath in response to that voltage. So this upper trace here is what it's called a trace, is depicting what you're doing, how you're manipulating the membrane of the cell, and the response of the cell is down here, and it's measuring the current going through one specific part of the membrane using an electrode. In this case, they have everything else blocked and they're looking only at sodium current. So what they show is that you have this sort of resting current, and all of these little blips, if you amplify this and look in this region, you'll see a big zigzaggy lines. And what that is sodium channels opening and closing, opening and closing, opening and closing, but not reaching what's called threshold, so that you get a big change in current. Over here, you see the opening and closing, which the membrane is doing because it's dynamic. The leak channels are changing constantly the voltage in one tiny region of the membrane. And since we're looking there, you'll see fluctuation of positive, negative, but only by a couple of millivolts. It's really tiny. When the movement is sufficient, it'll cause an action potential, which we'll talk about in a few minutes. But if I were to change the membrane potential to 30 millivolts to depolarize that membrane, what you see are sodium ions moving into the cell transiently, and then they stop moving, even though the cell is still depolarized, which means that the cell's sodium channels that are gated by voltage are opening in response to the depolarization, and then as the voltage begins to fall, they get gated, they get closed. So there are two different things that are happening with classic sodium channels that are voltage gated, where the voltage is causing movement of those charges inside of the channel to change the conformation of the channel and open it. This is that sort of open and shut door situation. And then a slower response to depolarization is the gating of this channel, and once it's gated, it then closes again, and you can see the membrane potential go back to its normal resting membrane potential. This voltage is being held at minus 50 millivolts, and here it's being held at minus 90 millivolts. This isn't a normal membrane that's just being allowed to do its own thing. By changing these voltages across the membrane, you're forcing it to do specific things, like activate voltage-gated channels, so you could see at what voltage those channels open. You can isolate the sodium channels by either using a toxin, usually, to close other types of channels, so that you know that the current that you're seeing is only caused by sodium. If you didn't block those other channels, there would be no way to tell that this current is sodium. We only know it's sodium because we've blocked the other channels in the membrane. So what happens when the potassium channels open? You guys constructed this in the last class. You get what is called an action potential. Potassium channels are also voltage gated, but they're slower. So the order of events is that the membrane is hanging out at minus 70 millivolts, and there's some stimulus that's going to open some sodium channels and depolarize that cell. Now that local small depolarization may or may not be large enough to open all of the sodium channels in that small region of the cell. In this case, the small depolarization was enough, and suddenly lots of sodium channels that are gated by voltage open. Now the other thing that happens here is potassium channels start to open also, but they do so in a little bit of a delayed manner. So what you see is a rapid depolarization of the membrane because sodium channels open. The sodium channels close at a depolarizing potential, and at the same time you start to see the potassium channels opening. This therefore causes potassium to equilibrate the membrane potential, and then it returns to resting when the potassium channels close. The potassium channels close right around here, but this return to the normal resting membrane potential is caused by the sodium-potassium ATPase. Now, again, reestablishing what should be happening, where you have sodium out, potassium in, and the leak channels are equilibrating roughly the potassium currents. Once one part of the axon is depolarized sufficiently to open up those sodium channels and cause a rapid depolarization and hyperpolarization, the sodium diffuses. So what this is depicting here is what's happening in one specific region of the axon. Now, in this particular region of the axon, there was some depolarizing event that opened up the sodium channels and caused a rapid and dramatic depolarization of the membrane. This depolarization spreads just because the ions are diffusing, and they may or may not be high enough a concentration to activate nearby sodium channels to thresholds so that they can also cause a rapid depolarizing event right in this region. And what this image is depicting is the direction through which the action potential is traveling down the axon. So you have a depolarizing event here, diffusion of sodium ions to neighboring regions, and then another depolarizing event. And it happens in rapid succession right down the axon so that something that starts here can travel all the way down to the end and cause changes at these synaptic terminals to give a message to muscle or to other neurons that need to receive that information. Now, here you should be wondering, or at least you should notice, that the diffusion of the ions goes forward in the correct direction, but it also goes backwards. So a question that you might get asked, or you might be asking yourself, is why doesn't this action potential that exists here in the middle of the axon not only go in this direction, but also go in this direction and mess up signals that are coming in from the dendrites intended to go this way? The reason for that is the inactivation of channels. So you have depolarization here that's going to cause more depolarization down the axon. Behind this is the repolarization caused by the potassium channels, and also inactivation of those channels, which causes a refractory period. And refractory means that when the action potential goes this way, the channels behind them aren't capable of opening again, or at least aren't capable of opening again to a sufficient extent to cause an action potential to start again. And this is really important for muscle movement and for essentially any neuron. You have to inactivate, otherwise you get something that's called tetanus, where there's just a constant stream of ions and it never repolarizes properly. So I can show you some experiments that I did in grad school next week where you can cause tetanus in a neuron where the neuron won't fire anymore because they're not repolarizing properly. Now, this action potential is not particularly efficient in the sense that having to cause all of these channels to open is a really long distance, and it's a lot to ask of a neuron. So if you think about walking from point A to point B, walking, if it's a long distance, can be a little bit tedious. But what if you skipped? When I bring my son to school, sometimes I trick him into rushing and I ask him to skip to school because skipping is, as it turns out, really fast. So if you skip, you would be making it all the way down here a lot faster than you would if you were just simply walking along. The way that the neuron can do that is by recruiting other cells called oligodendrocytes or Schwann cells to insulate this axon. And that insulation is called myelin. Myelin is one of the ways that you can make the propagation of an action potential from here at the axon hillock down to the end of the axon much faster. Another way to do it is to decrease the diameter of the axon. So a larger diameter of axon, when you have diffusing ions on the inside, is going to take a long time for those ions to coincidentally make it to the surface of the axon to activate more ion channels. Now this is Santiago Ramon y Cajal again. He saw this and predicted that these cells that were causing neuron to be created were really important to this neuron. He was wrong in thinking that they had to do with providing nutrition. But he did know that the myelin was being produced by cells in the central nervous system called oligodendrocytes. What he thought that wasn't right is that the oligodendrocytes were using portions of their cells to swing into synaptic clefts, to swing into the end of the axon, to close the axon so that people would sleep. Now that is not right. So although he was remarkable in his understanding of anatomy, physiology, he was a little bit weaker. So to make the action potential faster, when you have to send information from your spinal cord all the way down to the tip of your toe in a single axon, the strategy that's used in mammals is to decrease the diameter of the axon and also to add insulation or myelin. As I mentioned, oligodendrocytes make myelin in the central nervous system, and Schwann cells make it in the peripheral nervous system. Remember, the central nervous system is the brain, spinal cord, and the optic nerve and the retina. The peripheral nervous system is everything else. Now, the oligodendrocytes, because cells are so packed together in the brain and the spinal cord, are capable of myelinating more than one axon at a time. And what they do is they wrap around the axon at set intervals that are determined molecularly. We'll talk about that next week. And it creates these areas called nodes of Ranvier. And nodes of Ranvier are concentrations of ion channels. Now, if we think about what we would do if we were skipping down this axon and wanted to depolarize things really fast and get an action potential to propagate all the way down, you might guess that this region is packed with sodium channels. We want those sodium channels to be highly localized so that when you skip there, they're ready. And the voltage-gated channels are going to open up, let a lot of sodium in, and it's going to diffuse rapidly down a small axon and open them up here. The same thing is true for peripheral axons, but in the peripheral nervous system, Schwann cells produce that insulation, and generally a Schwann cell will create only one node. So they're also separated by a very specific distance and create nodes of Ranvier so that the signal can hop down this axon at very quick speeds. There are also unmyelinated axons, like pain axons. So axons that conduct pain are not myelinated. So, but you know that although your response is measurably delayed, you might have at some point touched a stove. It's remarkable that you see your hand on the stove and almost realize that you're going to get burned before you feel it. Because those axons are not myelinated, it's slower. That said, the response to pain is very fast. It's still fast. So you might imagine if action potential propagation can be made faster down an axon through the use of myelin and by decreasing the diameter of the axon itself, you might guess that pain neurons would have to be really, really tiny, and they are. The axons of these neurons are among the smallest in mammals. Because you still want them to be fast, but they happen not to be myelinated. And again, we'll look at this in more detail when we look at the spinal cord. So the myelin that's produced in the central nervous system and the peripheral nervous system is different depending on the location. Oligodendrocytes and Schwann cells are not the same cells. They serve the same function, but they express different proteins on their surfaces. The proteins on the surfaces of oligodendrocytes are highly inhibitory to any changes in the axon, where the myelin that's produced by Schwann cells in the peripheral nervous system are quite permissive to growth. This image is depicting the difference in propagation between an axon that's unmyelinated and an axon that is myelinated. This is an image from an electron microscope, and I think it's interesting because it shows in a good amount of detail what Santiago Ramon y Cajal saw, but this is a cross-section. So what you see in this upper left image is the axon itself here. In the solid arrow, these are microtubules. So the microtubules are the ones that go lengthwise along the axons. You can also see neurofilaments in the V. There's something called an inner tongue and an outer tongue, because these are the processes that wrap. Remember, they have ends. So you have one end that starts in and goes around, 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 and the cell ends up having another end with the nucleus typically embedded in here somewhere. In this image here, you can see the node depicted in N, as N. In red, these are the sodium channels, and the potassium channels are localized a little bit farther back. So they're diffuse. The membrane is covered in channels, but what this is showing you is the higher concentration of channels. Here, in the solid lines are the microtubules. You have a paranode, which is very close to the node, a juxtaparanode, and an internode. So you can see the layers developing as the oligodendrocyte continues to wrap around. These little lips are created here. So what happens when you close these channels? One of the ways that we can study neurons is by closing channels and seeing what happens. There are also situations in nature and in medicine where we want to close these channels. And these are exploited both in the lab and also in surgeries through anesthetics. So what happens when you block a channel? One of the things that's really common to look at is a compound called tetrodotoxin. Now, tetrodotoxin is a toxin that's produced by pufferfish. These are all pufferfish. One thing that you may not appreciate is that pufferfish don't always look like this. There are many, many hundreds of different types of pufferfish. So if you go to Florida and you see something that looks like this, be careful, because that's also a pufferfish that has tetrodotoxin. These toxins are also expressed by octopus and some other sea creatures. And you can see here that they actually do puff up. At the ends of these little spikes are where the tetrodotoxin is. And the fact that the fish has a toxin means that it doesn't have very many other defenses. This isn't a quick fish. It doesn't have teeth. It doesn't really have a way to protect itself. But the existence of the tetrodotoxin can paralyze and kill any of the predators that are trying to get it. Let's take a look at another voltage clamp example. Now, just to be clear, because there is a fair amount of text in the textbook about voltage clamping and patch clamping, the difference between those two is a voltage clamp is looking at a bigger region of the membrane. So it's taking into account many hundreds of channels and how they're contributing to the production of current across the membrane. Patch clamping is different because it's using a cannula, again, a piece of glass that you've pulled really, really, really thin, and you stick a piece of membrane to the end of that cannula. So you create sort of like a tent across the end of it. And the assumption is, because of the diameter of that cannula, that piece of membrane probably only contains one single channel. And then you can do single channel dynamics. You can look to see its opening and closing potentials, which is really important. And I guess potential is a little bit misleading. You'd want to say probability of opening. And also you can see what kind of compounds are going to shut that channel down or activate it more. So in this experiment, what we're doing is we're depolarizing the membrane from its resting membrane potential of, in this case, minus 65 millivolts to 0 millivolts. Now, when that happens, what you see is an inward current followed by a delayed transient outward current. So this trace, again, the trace is the line that depicts the current movement across the membrane in a specific region of the membrane. This is being produced by the movement of both sodium and potassium. Things are moving in, and then things are moving out. Now, the things that are moving in are depolarizing the membrane. This is the beginning of what would be an action potential. And then the things that are moving out are attempting to reestablish the resting membrane potential. So this is a little bit opposite to what we're used to seeing, because depolarization is this way, repolarization, and then return to resting membrane potential. But remember, when we're doing that, when we're looking at an action potential, we're looking at voltage. I is current. And what this is showing you is in arbitrary units, things moving into the cell and moving out of the cell. So when you're looking at these traces, the thing that you really need to pay attention to is whether it's depicting current, I, or membrane potential, VM. And that'll be the trick, hopefully, to you understanding what it is that you're looking at. So moving in are the sodium ions and moving out are the potassium ions. These are the things that are responsible for producing the action potential had this reached threshold. This is not sufficient to reach threshold, so you don't see a full action potential, but what you do see is activation of channels. And I want to emphasize that this brief depolarization right at this point is opening both sodium and potassium. But what you see are the sodium channels opening really fast because they're faster voltage-gated channels, closing because of that ball and chain inactivation. And then later on in here, you're starting to see the potassium channels opening and the potassium flowing out of the cell. This is getting activated at about the same time as the sodium channels, but it's delayed because the channels are just slower. If you use tetrodotoxin, you can isolate this potassium current. You can close the potassium channels and then you lose that inward current from, close the sodium channels, and then you lose the inward current produced by the sodium. And what remains is only the current that's being produced by potassium. And if you look at this relative to this trace up here, you can see that in fact, at the time that the membrane is depolarized, you also see opening of potassium channels. This is just lost in here because this inward current is so great, and it's mostly due to sodium. But if you were to close the potassium channels, what you'd see is it go down a little bit more. Because in this one, you have an average of both sodium moving in and potassium moving out. That's decreasing this curve just ever so slightly. In medicine, we can also use sodium channel blockers, and these are used for reasons that probably make sense. You want to stop somebody from moving. You want to stop them from feeling. Now, these specific compounds are closing specific channels. They're not closing every single sodium channel. They're closing some of them. So when you get lidocaine, the local anesthetic that the dentist might put on before they give you an injection so that you can have your cavity filled, that's going to close some of the sodium channels, and it's going to allow everything else to continue working except for propagation of pain back to your central nervous system so that you know that it hurts. It doesn't prevent the damage, but it prevents the signal of only pain. Now, when you have the injection that goes into the tissue itself, then you're closing even more channels. And that is a different compound that's causing not only the blockade of information about pain, but also information to the muscle that would cause it to stop moving. And that's really important not only for the dentist, but also for you, that you're completely numb. Numb meaning there's no information traveling to that tissue and no information traveling back. Potassium channel blockers usually are systemic, so isoflurane we use in the lab, chloroform we use in the lab, nitrous you might also get in the dentist. And what that does is it circulates through your bloodstream and again closes some of the potassium channels, but not all of them. So if you've ever had nitrous, hopefully just at the dentist, you'll know that you can still walk around, you can still talk, but you just feel a little bit loopy because those potassium channels that are being closed are not being completely closed systemically, only some of them are being closed. So today we've covered membrane potential in a little bit more detail. We've also covered the effect of myelin and also blocking some of those channels using toxins. Please email me if you have any questions about the Canvas quiz or any questions about this lecture at all, and I'll be happy to email you back as soon as I can.